Hello everyone, welcome to my channel again. So in my last video I discussed about mutation, that is gene mutation. So as we know that mutation can be gene mutation or it can be chromosomal mutation. So in this lecture I am discussing about the chromosomal mutation. So chromosomal mutation is also called as chromosomal aberration. So chromosomal aberration means any kind of change that takes place in the chromosomes. Like in the gene mutation, the change takes place in the DNA. That can be also called as point mutation. If you remember my last video, there the change occurs in a particular gene, in a single gene. So a change comes in a single gene and that will alter the phenotype of the organism. So you can imagine that if a single change, single base pair change in the single gene, if it occurs and if it changes the organism's phenotype, then you can just imagine if the change takes place in the chromosome, what kind of change will take place. Like the chromosome, they contain the gene, like this is the structure of a chromosome and the chromosome contains gene in it. So if the change takes place in the number of the chromosome or if it takes place in the structure of the chromosome, then not only a single gene, many genes will be affected. Many genes will be affected, and if many genes will be affected, then the problem will be more. Okay. So here let us go. Chromosomal mutation, it can be of two types. It can be structural changes in the chromosome, in which the structure of the chromosome gets changed. See, we have, if we say about the human being, 46 number of chromosome, that means 23 pairs, and the structure of the chromosome is like this. This structure is very specific. You cannot change any kind of change, you cannot bring any kind of change in the structure. Like if this structure of the chromosome becomes like this and this part of the chromosome is deleted or it something happens to it, then this chromosome is a mutated chromosome, it will create some phenotypic change. And same thing happens in the numerical changes of chromosome. In numerical changes of chromosome, the structure of the chromosome is almost same, there is no change in the structure, but the number of chromosome will be changed. What kind of change? If the number of chromosome is 46, then it can be 47, as we discussed in our last video, that is genetic disease. It can be 48 or it can be 46, it can be less or more. But in numerical change, the number of structural change, the number of chromosome remains the same, 46, but the structure of some chromosome or any one chromosome, it may be changed. So it can be structural change or it can be number, that is numerical change of the chromosome. Now let us move to the structural change in the chromosome. The structural change in the chromosome may occur by this following mechanism. There are different types of mechanism, like deletion. What happens in deletion? Say for example, there is a chromosome and this chromosome contains some gene A, B, C, D, E, F. These are the gene in this chromosome. If any change occurs in this chromosome and if deletion occurs, say for example this AB, this portion of the chromosome containing AB gene will be deleted, then the resulting chromosome will be like this, having gene C, then D, E, F. If it happens, then that chromosome will be called as deleted chromosome. That means this A and B segment, this segment of the chromosome containing A and B gene is deleted and it is lost. This is also called as deficiency. And I told you about this problem, that is in chromosome number 5. If the small arm of the chromosome number 5 is deleted, then it will create a disease called Cry-Do-Chart syndrome, and I have told about it. This is what deletion. There is one more thing, that is duplication. Duplication can also take place. What happens in duplication? If the chromosome is like this, the gene is 1, 2, 3, 4, 5. If this is the structure of the normal chromosome, then this part will be duplicated. How it happens? It will be duplicated here only and will be attached to this homologous region. That means 1, 2, then again 1, 2, 3, then 4, 5. That means the new chromosome is containing 1, 2, 3, 1, 2, 1, 2, 3. That means 1, 2 gene is duplicated here. That means first 1, 2 this duplicated region, and again the normal region 1, 2, 3, then 4, 5. This will also create some problem in the organism. So, this is the second type of structural changes, that is duplication. Now, one more is there, that is called inversion. In inversion, there is no loss or no gain of the chromosome or the gene. There is no loss or the no gain, but what happens in inversion, the the placement of the gene is altered, that is their places, the specific location of the gene is altered. Say for example, this is a chromosome having a number of gene here, and these gene are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. If this chromosome alters its gene, how? It rotates by 180 degree like this. So, the chromosome, the arrangement of gene is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and the place where it alters, where it rotates, the place where it rotates, it will cut itself from here and rejoined. As a result, this portion will be joined to here and this portion will be joined to here. The resulting chromosome will be having the gene in a sequence that will be altered 1, 2, then 3 was in this part 3, then this will be here 7, 6, 5, 4, then this will be attached to this 8, 9, 10. So, here you can see the 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, this sequence of gene is altered here 1, 2, 3, 7, 6, 5, 4, 8, 9, 10. This, there is no change in the number of gene, but the arrangement of gene is changed. If this arrangement of gene gets changed, this chromosome will also be called as mutated chromosome, because it will be having trouble in the crossing over. See, in inversion there are two types. It can be pericentric inversion, or it can be paracentric. So, in pericentric inversion what happens? If this alteration occurs, then the centromere of the chromosome is affected. Centromere, you know that in the chromosome there is a centromere. This position of the centromere get affected. If this position of the centromere get affected, it moves from one position to another position in the resulting chromosome. That means the inversion involves centromere, where the position of the centromere is replaced or it is altered, that is called pericentric. And if the paracentric inversion takes place, then the centromere position will be there only in the original position. The part of the chromosome that will be altered, it will be far from the centromere, and the center of the position of the centromere will not be altered. That kind of inversion is called paracentric. So, in pericentric the centromere is involved, its position is altered. In paracentric the centromere is not involved, its position is not altered. This is what inversion. Now, come to the next type, that is called translocation. What happens in translocation? It is just the deletion and the addition of the chromosome, and that is to non-homologous chromosome. What happens if there is a chromosome having a gene A, B, C, D, E, F? This is a chromosome, and there is one more non-homologous chromosome. Non-homologous chromosome means it is not of the same pair, it is some another chromosome, and the gene here is 1, 2, 3, 4, 5, 6. Now, what happens? This part of the chromosome will be deleted, and this will be going and attaching itself to some non-homologous part. As a result, this chromosome will become like this. It will be having only C, D, E, F, but the other chromosome, non-homologous chromosome, any other chromosome that can be having that A, B, then 1, 2, 3, and then 4, 5, 6. That means the part of this chromosome attached to the non-homologous chromosome, that is called translocation. The translocation can be reciprocal translocation or simple translocation. This example that I have given to you is a simple translocation. From one chromosome the part is transferred to another chromosome, and where there is no exchange, just here the deletion occurs and here the addition occurs. But in reciprocal translocation what happens, the part will exchange their segment. How? Let me give you one example. If the chromosome is this, having A, B, C, D, E, F gene, and another chromosome is there which is having the gene 1, 2, 3, 4, 5, they will exchange their part. What happens? This is the non-homologous, mind it. If the chromosomes are homologous and they exchange their part, this is crossing over. But if the chromosomes are non-homologous and they will exchange their part, this is not normal, this is reciprocal translocation. What will happen to here? This chromosome will become, this part will be transferred to this chromosome and this part will be transferred to this chromosome. That means this chromosome will be 1, 2, then A, B, C, D, E, F, and this chromosome will become A, B, this part get attached to this, and A, B, after A, B 1, 2, 3, then 4, 5. So, this kind of change, if this kind of change occurs, this is called reciprocal translocation. So, this is about the structural changes in chromosome that we got: deletion, duplication, inversion and translocation. They all will cause some disease in the organism. Now, come to the numerical change in chromosome. Numerical change in chromosome is something different, where the number of the chromosome get altered. How the number changes? This can be euploidy or this can be aneuploidy. What happens in euploidy? In euploidy, the whole set of chromosome will be changed. You know, whole set of chromosome, that means there is a set of chromosome. Every organism has a genome, and the set of that is the haploid set, and the haploid set of chromosome is called genome, or we call it N. We represent it like N or X, whatever. So, this is the whole basic set of a chromosome. We, the human being, we are diploid, we have twice N, okay, or twice X. In euploidy what happens? The whole set of the chromosome is changed. The whole set of the chromosome is changed. This is not totally natural. Naturally it takes place, but it is actually induced artificially, and it is basically induced in case of plant, because after we make polyploid, then we can get various different type of character in the plant, like the size of the fruit, nutrition in the fruit, the specific nutritive substance in the fruit, or in the food of that plant material can be increased by inducing euploidy, by making them euploid or polyploid. So, how it takes place? Monoploid. Monoploid are the organism where the chromosome number is X, that is X or N, whatever. So, say for example honeybee. In honeybee you can get the monoploid, the drones. The drones are the haploid having a single set of chromosome, single genome. But what are the diploid? Diploid are the organism where you get the double. So, all the organism in this earth, almost all, not all actually, almost maximum organism are diploid. Like we human being, we have twice N. So, our basic set is N, where the chromosome number has been doubled and it becomes diploid. Okay. So, this is what diploidy. Now, there is one more, polyploidy. If the chromosome number doubles to become thrice N, if it becomes double to become 4N, 5N, 6N, then this is called triploidy, tetraploidy, pentaploidy, hexaploidy, heptaploidy and so on. As a whole they are called polyploidy. Okay. And this polyploidy is not seen in the animals, this polyploidy is generally seen in the plants, and in plants the polyploid is induced, and if it is induced, then some character of the plant can be changed, which can be beneficial for us. The polyploidy can be autoflow polyploidy or the Alo Polyploidy. What happens in the autoflow polyploidy? In the autoflow polyploidy, same species, the chromosome number will be changed of the same species. Say for example, one species is having chromosome number AA. This is the normal diploid organism, and if we induce polyploidy here, the chromosome number can become AAA triploid, or we induce more, it can become AAAA tetraploid. This is polyploidy. The number of chromosome, the set of the chromosome will be changed, but the set will be of the same species within the species. Okay. One more is there, Alo Polyploidy. Alo Polyploidy occurs when the chromosomes are from the different species. Say for example, you take one species having AA, you take another species having BB, and generally the interspecific hybridization is not possible, but in case of plant it can be done. So if you do it and you cross these two plant, these two individual, then you get AB, and this is the genotype of the organism. This is generally you can say this is sterile. In case of animals also it can be done, that the mule is the interspecific hybridization by the hybridization, that is by the fusion of horse and donkey. Now this AB is a sterile, but if you can induce polyploidy here, it can become AABB. So you get the 4 set of chromosome, and this can be called as tetrapolyploid, and this tetrapolyploid is not autopolyploid, because this is the combination of two species. There are the chromosomes of the two species mixed together. This is called Alo Polyploidy. But if you do it in the same species without mixing it with the other species, this is called autopolyploidy, and this one is Alo Polyploidy. So this can be done. There is a chemical called colchicine, and this chemical is highly useful in inducing polyploidy in case of plant. So in this case we use the colchicine. This is also a mutagen. So now come to the aneuploid. So if you remember that the numerical changes are two type, euploid and aneuploid. Euploid are the change in the whole set of the chromosome, but the aneuploid is not about the whole set of the chromosome. Aneuploid is the change in a particular pair of the chromosome. Say for example, I take the example of human. Humans have 46 number of chromosome, and this 46 number of chromosomes are actually 23 pair. So there is one pair, there is second pair, there is third pair, there is fourth pair, and every pair consists of 2 chromosome. If the change occurs in any pair of the chromosome, not the whole set, mind it, in euploid the whole set of the chromosome is increased, but in aneuploid any pair of the chromosome is increased. Say for example, any individual is there who is having 3 chromosome in the first set, and other rest all the chromosomes are normal, then that individual will be having 46 plus 1. What is that 1? That extra chromosome is present in the first pair. The first pair should be having only 2 chromosome, but here there are 3 chromosome. This is called aneuploid. That is, aneuploid takes place in any of the pair of the chromosome. That you can say that if there is a change in any pair, any homologous pair of the chromosome, that kind of change is called aneuploid. So that change can be one more, or that can be less. That is, here there are 3 chromosome in the same chromosome number pair 1, but it can be only 1 chromosome in any pair. So the chromosome number can be increased or the chromosome number can be decreased. If the chromosome number is increased, that is 1 or 2 chromosome will be increased in any pair, that will be called as hyperpolyploidy. And you know the meaning of hyper means more. And if the chromosome number is decreased from any pair, like any pair is there where there is no 2 chromosome, there is only a single chromosome in this pair. Say for example, pair number 20, there is only a single chromosome, the other pair is lost. If this occurs, that means there is a loss, there is a less number of chromosome, that is called hypoploidy. Hypo means less. So this aneuploidy are of 2 type. It can be hypoploidy or hyperploidy. Hypoploidy, less number of chromosome will be there in any pair. Hyperploidy, more number of chromosome will be there in any pair. Now you see, this hypoploidy can be monosomy and nullisomy. Mono means 1, and nulli means no, 0, means in any pair there is only one chromosome in that individual, and you have got it in genetic disease. I suggest you to watch my video on genetic disease. There you will get the problem that will cause due to the monoploid or trisomy or tetrasomy. I have explained there. So in monosomy there is only one chromosome in any of the pair. In nullisomy there is no chromosome in any of the pair, it is totally empty. Trisomy, I told you, in trisomy 21, that is Down syndrome. What happens? One more chromosome is there in the 21 number of chromosome. So that is one example. So if one more chromosome is there in any of the pair, then it becomes twice n plus 1, and if two more chromosome will be there in any of the set, then it will be twice n plus 2. Twice n is the basic set. If it is hyperploidy, then plus 1, or it can be plus 2, or if it is hypoploidy, it can be minus 1 or minus 2. So let me give you how it occurs. It occurs due to chromosome nondisjunction. This occurs due to chromosome nondisjunction. What is nondisjunction? Nondisjunction occurs when there is a process, that is meiosis. In meiosis cell division, the same number of chromosomes are not transferred to the gamete. If it happens, because we know that the chromosome number is 46 and it forms 4 gamete. So in the gamete we should have 23. So say for example, a male is there having 46 number of chromosome, a female is there having 46 number of chromosome. If it forms if all the pair of the chromosome are not divided equally, then it can form 22 and it can form 24. And if here it is normal 23 and 23, then if this gamete fuse, then 22 plus 23 equal to 45, that is one chromosome is less. And if these two gamete fuse having 24 and 23, this will be 20 or 47, that is one more. Now again I will explain it in detail. Say for example, 46 chromosome in male and female. Say for example, this will have two gamete, and the exchange of the chromosome or the separation of the chromosome is not normal. Say for example, chromosome number 1 is here, 1 comes here, second comes here, second comes here, third comes here, third comes here. But in case of number 4 chromosome, nothing comes here, 0, and both comes here. So as a result, chromosome number 4 will be empty here. So it will be having 23 and it will be having 24 number of chromosome, because in chromosome number 4 both the chromosome came here. Now if it forms a normal gamete, if this is female and this is male, if this male forms the normal gamete 1 2 3 4 5 6, 1 from each pair, it will be having 23 and it will also be having 23. If this and this meet, then what will happen? Chromosome number 1 from this, chromosome number 1 from this, they will have a pair. 2 will be having pair, 3 will be having pair, but in case of 4 you will get 2 from here and 1 from here, 3. That means in chromosome number 4 you will get 3 chromosomes. That means this organism will be having, this individual will be having 46 plus 1. This is what trisomy. What is tetrasomy? Say for example, this is hyperploidy. What is tetrasomy? Say for example, this is female having 46, this is male having 46. This will form gamete having 22 and 24. This will form gamete having 22 and 24. Same problem occurred both in male and female. And this 24 if they fuse, say for example the 24 number of chromosome occurred due to the 4 number, due to the both the chromosome came in the 4 number chromosome, here also in the 4 number chromosome, then what will happen? Chromosome number 1 will be having, chromosome number 2 will be having 1 pair, 3 will be having 1 pair, but chromosome number 4 will be having 2 from here, 2 from here, 1 2 3 4 chromosome. That means the tetrasomy. This is the case where tetrasomy occurs. That means this individual will be having 46 plus extra 2. Okay, so now let us discuss about monosomy. I have discussed about monosomy. Nullisomy, say for example this is male, this is female. This male will form gamete having 22 and this one chromosome is less in the chromosome number 1, say for example, another is 24. This female exact it does the same, 22 chromosome and 24 chromosome in the gamete, and this 22 chromosome, because one chromosome is less here also in chromosome number 1. So when these two fuses, what will happen? It will not give any chromosome in the chromosome number 1 empty, and it will also not give in any chromosome in the chromosome number 1 empty, but the rest are normal. 2 number chromosome 1 pair, 3 number chromosome 1 pair, 4 number chromosome 1 pair. So this zygote will be having no chromosome of pair number 1. So that means here 46 minus 2, that means there is no chromosome here, that is called nullisomy. And if this problem occurs only in the single parent, say for example it is having 23 and it is having 22, then there will be one chromosome here, this is called monosomy. So this is about the chromosomal aberration. So if you have any doubt, please ask in the comment below. So this is about the chromosomal aberration, and here I complete the mutation part, that is totally the mutation, that is gene mutation and the chromosomal mutation. Thank you for watching my channel. Thank you so much.